Hello everyone! In this video we'll be explaining to you the affinity chromatography general concepts and protein AG as an example. We are Ji-yeon, Farzad, and Salma.
I'm gonna explain you about the history of affinity chromatography. So have you ever thought how to selectively separate your product after the production step of a bioprocess? Well, consider affinity chromatography as a great strategy. So I'm gonna tell you an interesting story about how affinity chromatography got to become so familiar with us these days. Between 1950s and 1960s, there were several original researches describing isolation of enzymes by ligands of selective chromatography steps. One example is the use of flavin cellulose compounds in column chromatography to purify liver flavokinase. In 1968, the term affinity chromatography was first used in a seminal article written by these two, Pedro Quatro Casas and Meir Wilczek, who both later got Wolf Prize in Medicine in 1987 for their contribution for invention of affinity chromatography and its applications to biomedical sciences. In the 1970s, various ligands capable of interacting with several targets were introduced. The examples are protein A, coenzymes, lectins, nucleic acids, metal chelates, and etc. Around the same time, hybridoma technology were also developed to produce monoclonal antibodies, which started the wide use of monoclonal antibodies in biomedical separations.
Like all other chromatography techniques, the affinity chromatography is composed of similar instrumental components too. The first part is the solvent or the mobile phase, which is selected based on the nature of the sample. The second part is the column or the stationary phase. Column is filled with a matrix material called resin. In the last part, we have the detector. Different types of detectors like ultraviolet, infrared, refractive index, electrochemical, and mass spectrometer detectors can be used. In all chromatography techniques, including affinity chromatography, samples are transferred with the mobile phase either manually in gravity-based systems or with the help of a high-pressure pump in automatic systems to the column. As sample molecules are separated based on their specific properties and their interaction with the resin inside the column, they are detected by a detector and accurate data appears as a chromatogram. In affinity chromatography, the general separation principle is based on a highly specific macromolecular binding interaction between the biomolecule and another substance. This interaction can be between an antibody and antigen, an enzyme and substrate, or between a receptor and ligand. Different types of binding interaction commonly used in affinity chromatography procedures are summarized in this table. In general, all types of affinity chromatographies are composed of three steps. After column equilibration with the mobile phase, in the loading step, a mixture of different molecules is passed through the column and the binding partner on the resin captures the molecule with specific affinity. Non-targeted compounds are washed away in the wash step. And finally, in the elution step, a buffer with different ionic strength or pH will disrupt the interaction and releases the molecule of interest from its ligand as a purified product. One of the best examples of the affinity chromatography techniques is the use of protein A for purification of monoclonal antibodies. Protein A is a surface protein from a gram-positive bacteria, Staphylococcus aureus, and is able to bind specifically with strong affinity to the Fc portion of the immunoglobulin molecules. Therefore, by leveraging this unique feature, protein A can be used as the capturing molecule for purification of antibodies which are produced inside the laboratory.
The next part is about manufacturers. There are many manufacturers providing protein A and G products for antibody purification, such as Thermo Fisher, BMake, Santa Cruz, Biorad, Millipore Sigma, and Abcam. For example, Thermo Fisher provides different kinds of protein A and G products like magnetic beads, magnetic agarose beads, and agarose, and they are based on their different properties like bead or resin size and their binding capacity toward different antibodies. Next part is the versatility of affinity chromatography. So affinity chromatography can be used in many different stages in downstream process. These include the capturing steps and polishing steps. So affinity chromatography is mainly used to capture the target molecules from solution of mixtures as seen in this picture. However, it can also be used in polishing steps, such as removal of process-related impurities when used in flow-through mode. The resins in this case are called scavenging affinity resins, or they can be used to separate the desired form of a target molecule from closely related product impurities by, for instance, selective elution. Most common types used in affinity chromatography, protein A and G affinity chromatography, and histidine tagged affinity chromatography show common yield of 50% and 90% each for single step, which can be lower or higher with different operation conditions.
The uses of affinity chromatography mainly include separations, purifications, analysis, or characterizations. Many of the applications involve both small and large-scale purification of biomolecules, such as enzymes, native and recombinant proteins, by using dye-ligand or biomimetic affinity chromatography. Common examples of popular small-scale applications are isolation of histag proteins by immobilized metal affinity chromatography and isolation of specific antibodies or antigens by immunoaffinity chromatography. However, this technique could also be used for other applications, such as cell affinity chromatography, to isolate certain types of cells by using interactions between cell surface agents and their ligands. It is also used for analytical methods for different purposes, such as separations or analysis of chemicals in samples, sample pretreatment prior to further analysis, or chiral separations important for drug discovery and development. Also for the examination of biological interactions to characterize their binding strength, their kinetics, and the number or types of sites that are involved, for example between proteins or drug protein, enzyme inhibitor, antibody antigen, etc. Finally, it could be used for indirect detection and measurement of a target analyte by chromatographic immunoassays.
In affinity chromatography, volume of the sample that can be loaded on a column directly depends on the resin capacity and size of the column. This value varies from milliliters in small scales to cubic meters in industrial scales. Many different types of column with different sizes are available in the market. Based on the dimensions and some resin factors like mechanical and chemical stabilities, flow characteristics, and the most important one, based on their binding capacities, they can cost between few hundreds to several $1,000s. It is good to be mentioned to pack more quantities of resin to increase the overall volume in the large-scale column categories in order to keep similar linear velocity of the fluid inside the column. The height of the column tends to be kept same. Therefore, the increase will be in column's diameter. By keeping the height and flow rate same, the performance of the resin will be maintained equal in different scales, and depending on the starting material quantities, different yields from milligrams to multiple grams can be obtained.
Now, the purification step in a downstream processing scheme can be also performed with other technologies. For instance, with ion exchange chromatography, where the separation is based on electrostatic affinities. This method can sometimes be a good alternative to purify monoclonal antibodies of a basic isoelectric point, because the cost is almost one-fifth compared to protein A chromatography. Another alternative format is membrane chromatography, which combines the advantage of membrane filtration with liquid chromatography. The adsorptive membranes used here have shorter diffusion times than those obtained in bead chromatography, as the interactions between molecules and active sites on the membrane occur in convective through pores rather than in stagnant fluid inside the pores of an adsorbent particle, as we can see here in the figure. Therefore, this technique is cost-effective to purify large biomolecules at high flow rates, reducing biomolecule degradation and denaturation. This is why many innovations have been recently attempted here, such as increasing the binding capacity with hydrogels and nanofiber-based matrices. Also, there's annular continuous chromatography, where the chromatography bed is packed into a rotating annulus to work in a continuous mode. It has been used to purify recombinant blood clotting factor IX, virus vaccines, and recombinant antibodies from the cell culture supernatant. Finally, there's multi-column countercurrent chromatography, where at least 3 columns are required. The first one is loaded to saturation. The breakthrough is loaded onto the next columns, and the saturated column is washed and eluted as in a batch operation. This one is useful for resolving various protein variants, with high yield and purity of the target product, like is the case of commercially available recombinant streptokinase, single-chain antibody fragments, and various monoclonal antibodies. The first advantage of affinity chromatography is that it reduces the number of purification steps, which in turn increases the overall process yield and reduces processing time. In this figure, we can see the comparison of non-affinity and affinity-based purification processes. Even if the two first non-affinity steps have much higher loading capacities, using affinity chromatography as the first step can reduce the overall cost of goods 1.5-fold as compared to the non-affinity process. This is because the first non-affinity step has a much lower yield, as we can see here. Thus, the overall process yield ends up being significantly lower. Also, cost of goods can be further reduced by increasing resin cycling and improving target loading. For instance, protein A resins are now routinely cycled between 100 to 200 times. Now, affinity chromatography also allows for standardization in different cases, for example, through platformization. This refers to a highly selective affinity chromatography-based capture step, followed by a few polishing steps. Typically, each company develops a few variants of a platform process with different unit operations post the capture step, in order to properly purify variants of a target molecule. So in addition to standardizing manufacturing operations, this concept plays a vital role in scale-up and technology transfer, as well as in defining layouts of manufacturing facilities. It also shortens timelines and reduces cost of process development, because a very robust downstream purification process can be developed and scale up with a lower number of full-time equivalents. Finally, this standardization also enables a continuous and intensified mode of operation. Now, the particular advantages of protein A affinity chromatography include its simplicity and large purification factor, but also the specificity, because it relies on a very specific interaction that is not sensitive to variations in the feed stream composition in comparison to other techniques. Also, because of its high conformational stability, protein A is able to refold after denaturation, it resists cleaning with reducing agents, it has a fair degree of steric mobility, and its binding domains are protease resistant. Finally, this step is great for volume reduction since it provides a 5 to 10-fold concentration of the product.
Despite all benefits of affinity chromatography techniques, there are some limitations too. First one, it takes a lot of skill to handle it. Affinity chromatography requires some good technical skills to perform effectively and sometimes can be tedious and time-consuming, especially in the case of large-scale samples. The second limitation relates to the sample volume, which directly depends on the binding capacity of the column. Also, the ligands used in affinity chromatography are costly. Overall, the demand for specific ligands make the process an expensive technique. Ligand leakage from the column is another problem which directly affects the product's purity as well as column efficacy. Moreover, non-specific adsorption can be observed in some affinity chromatography approaches. And finally, using extremely low or high pH solvent and buffers will be resulted in degradation of the solid supports and limits the resin lifetime.